Alright, so porin is my protein, so let's go ahead and get started so this presentation isn't entirely too long. Okay, some general background information about proteins. Proteins have very diverse functions, and some of those functions include like transport proteins, structural proteins, and ones used for movement. Amino acids are linked by the peptide bonds, and you might be like, oh, what's an amino acid? Well, amino acids basically form and hold together proteins. And there's 4 levels of structure: the primary, secondary, tertiary, and the quaternary. And the primary is a unique amino acid sequence, and it just basically describes what order the what order the amino acids are in order to create the specific protein we need. And then the secondary is a conformation of amino acids, and basically here you see the beta sheets and the alpha helices. And then the 3D then whenever they start to fold on each other, you'll begin to see the tertiary, which is a 3D structure, and then quaternary is whenever you have multiple polypeptides. Some background information that I stated in the little article thing we did. Porin was originally discovered by Hiroshi Nikaido. Okay, so here we have a picture of porin. So these little gray thingies in the middle, that's actually a porin molecule, and it's located on the outer membrane of the gram-negative bacteria. And essentially it takes the stuff from the outside of the cell into the periplasmic space. So this is the outside of the cell, this is like the outer membrane, and it moves it right here into the periplasmic space. And it basically just facilitates the diffusion of like small molecules and stuff to help make sure everything really flows through. Okay, next slide. Some of the functions. It functions basically, as I said before, a transport protein, and it allows ions and small molecules to diffuse into the periplasm from the external environment. And it's embedded in the phospholipid bilayer, which makes it like easier to diffuse things. And it forms a water-filled pore from the external membrane to the periplasm, and it does this by diffusion. And diffusion is passive transport. So passive transport is easily described as like basically like laid back, lazy, no energy. So you don't need no energy to do this, so basically it's done in all bacteria. That's why it's so important. And it allows cells to regulate the internal environment, because if things don't go in and out, then it could like swell or shrivel, so you got to make sure there's good movement. And it allows diffusion of small hydrophilic molecules into the periplasm through a pore, that's a typo, through a pore, and the pore of 3 beta barrels. And basically the pores are in the center, and each beta barrel that makes up porin. Okay, so for the structure, there's roughly 301 to 420 amino acids long. It depends on the specific porin molecule. And there's roughly 274 solvent molecules, 3 calcium ions, 3 detergent molecules, and an unknown ligand. So this is the primary structure for the Rhodobacter capsulatus. Trypsin, I can't say these words, but in protease and carboxyl peptidase. I have a hard time saying these things, sorry. And that's a picture of the Rhodobacter, and that's basically the primary structure. Those are the things that are inside of it. The secondary structure, it has anti-parallel beta pleated sheet backbone. So it's not exactly like this one that you see in the first one. These are all nice and smooth and they don't fold back each other. That's not what the porin really does. These ones like fold back on each other because they're anti-parallel, like in this picture. So this one's like not what it is, that's actually what it is. And a fun fact, porin was the first transmembrane protein for which beta structure was established equivocally. And essentially, this picture right here is showing that they fold back on themselves and they're not smooth like A. So for the tertiary, it's always anti-parallel. This is actually what the beta barrels look like that I was referring to and continue to refer to. Like I said, they're always anti-parallel, and basically it's when the beta pleated sheets come back on each other and they form like a little loop. Like if you look at it from the top, you'll see a little loop or whatever. And then it like comes down and it makes like a barrel. And we refer to these as beta barrels. And basically the pore comes down in the center if you're like looking from the top, and it forms a barrel, which I already said that. So the quaternary structure, it has 3 polypeptides linked together, and each each is a beta barrel, and they're linked together, and each has a pore in the center as well as a pore in between the polypeptides. So this is a beta barrel, that's a beta barrel, and that's a beta barrel. And in the inside, it's polypeptides, and there's a pore in like these three things, these three beta barrels, sorry, and in the middle that like cluster that holds them together. And since they're all linked together, they can diffuse into one another easily. And 3 beta barrel subunits have 1 pore each. So the structure, it reverses the usual pattern. Usually the hydrophobic residues are on the interior, but polar residues make it on the outside. And Porin's beta barrel has an opposite makeup, usually, because the polar hydrophilic residues are on the inside, and the hydrophobic residues like flip on the outside. And this is because Porin's in the plasma membrane, and it has to attach. So it has to be opposite in order to like cling to it. So the hydrophobic and the hydrophilic regions of the beta barrel like structures have to flip. And attracting surrounding lipids in the phospholipid bilayer of the plasma membrane, of which Porin is a part. So in conclusion, Porin's essential to all bacteria because it deals with the transmembrane transport, because it's on the outside, the outer membrane, and like helps facilitate and diffuse like small molecules in and out of the cell, because without it, it could cause some serious damage. And it basically moves moves molecules from the periplasmic space to the external environment, and the complex structure essentially helps bacteria's function. And those are some of my sources, so that's it.